Welcome to Science Buzz. Today we're going to do a deep dive into something pretty fascinating. Cellular respiration. You know, that process that turns the food we eat into the energy that keeps us going. It's kind of like our cells have their own little power plants, humming away 24/7.
Yeah, it really is remarkable when you stop and think about it. We take a bite of food, a sandwich or whatever,
Yeah.
and our bodies somehow convert those carbs and fats and proteins into fuel. That powers our muscles and our brains. Like, how does that even work?
Right. It's amazing. So how do you really do it? That's what we're here to figure out. So let's start with the basics. What exactly is cellular respiration?
Well, it's basically this carefully choreographed energy conversion process. You can imagine each cell as a bustling city with tiny power plants called mitochondria.
Okay, I'm picturing these microscopic cities with tiny power plants. What happens inside those power plants?
Well, those mitochondria are the sites of cellular respiration. They take in fuel, mainly glucose from the food we eat, and convert it into a usable form of energy called ATP, adenosine triphosphate.
ATP, I've heard of that. It's like the energy currency of cells, right?
Exactly, just like we use dollars to buy things. Cells use ATP to power all their activities, from building new molecules to transporting things around.
So how does ATP actually work? What makes it so energetic?
The secret lies in its structure. Imagine ATP as this tiny molecule with the tail made of 3 phosphate groups. And these phosphate groups are negatively charged. They really don't like being near each other.
Oh, so they're like magnets repelling each other.
Exactly. And that creates a lot of potential energy, like a tightly wound spring. When the cell needs energy, it breaks the bond between those phosphate groups and bam, energy is released.
That's a pretty ingenious system. So cellular respiration is all about generating this ATP energy currency. But how does that process actually happen?
Well, it's a multi-step process, a bit like an assembly line with different stations. We can break it down into 3 main stages: glycolysis, the Krebs cycle, and oxidative phosphorylation.
Okay, 3 stages. Let's start with glycolysis. What's going on there?
Glycolysis is the first step, and it takes place in the cytoplasm of the cell, not inside the mitochondria. Picture this. You take a bite of a sandwich, your digestive system breaks down those carbs into glucose, and that glucose makes its way to your cells.
So the glucose arrives at the cell. What happens next?
That's when glycolysis kicks in. It's a series of reactions that split that glucose molecule, a 6-carbon sugar, into 2 smaller molecules called pyruvate, each with 3 carbons.
So it's like cutting a log in half. Does it take energy to do that splitting?
Yeah, you're right, it does. It takes a little bit of ATP to get glycolysis going, like an initial investment, like putting a coin in a vending machine. But the good news is that glycolysis actually generates a bit of ATP in the process, giving you a net gain.
So it's an energy investment that pays off. What happens to those pyruvate molecules? Where do they go next?
That's where we head into the mitochondria, our cellular power plants. And into the second stage, the Krebs cycle.
Ooh, I'm ready for a tour of these power plants. Can you describe what they look like?
Picture a tiny bean-shaped structure with 2 membranes. The outer membrane is smooth, but the inner membrane is folded into these intricate structures called cristae.
Cristae, what are those for?
They're like shelves, increasing the surface area of the inner membrane. This provides more space for all the amazing reactions of cellular respiration. And within that inner membrane is a space called the matrix. That's where the Krebs cycle takes place.
So our pyruvate molecules arrive in the matrix of the mitochondria. What happens to them?
They undergo a transformation. Each pyruvate is converted into a 2-carbon molecule called acetyl-CoA, which is the key to entering the Krebs cycle.
Acetyl-CoA. Got it. So this is where the real energy production starts to ramp up.
Exactly. The Krebs cycle is a series of chemical reactions that methodically dismantle that acetyl-CoA molecule.
Dismantle it. Why would they do that?
To release its energy, of course. But not all at once. It's a controlled demolition. The energy is captured and stored in the form of high-energy electrons, which are carried by molecules called NADH and FADH2. These are like energy vouchers that will be cashed in later for even more ATP.
Energy vouchers. That's a great way to put it. It's like earning points on your credit card to use later.
Yeah.
But hold on. What about organisms that don't have mitochondria? Do they just miss out on all this energy production?
That's a great question. And it leads us to a fascinating side path: fermentation. Some organisms, like yeast, can't do the Krebs cycle because they lack mitochondria. Instead, they rely on fermentation to generate energy.
Fermentation. Isn't that how we get beer and bread?
It is, but it also happens in our own bodies. Think about those times when you've pushed your muscles really hard, like during a tough workout.
Yeah, like that burning feeling in your legs when you sprint. What's going on there?
That's lactic acid fermentation. When your muscles are working intensely and don't get enough oxygen, they switch to this backup energy system. It's not as efficient as the Krebs cycle, but it's a way for your cells to keep generating ATP when oxygen is limited.
Well, so our bodies have a backup energy system. That's pretty cool. So we've got glycolysis breaking down glucose and generating a little ATP. And then the Krebs cycle in the mitochondria generating energy vouchers in the form of NADH and FADH2. What happens next?
That's where we head to the grand finale of cellular respiration, where those energy vouchers get cashed in for a big ATP payoff. And that happens in the third stage, oxidative phosphorylation. We'll dive into that in part 2 of our deep dive. Stay tuned.
Welcome back to Science Buzz. We're continuing our exploration of cellular respiration, that incredible process that turns food into energy. Last time we left off with a stack of energy vouchers in the form of NADH and FADH2. So how do those vouchers get cashed in for some serious ATP?
That's where the most fascinating part of cellular respiration happens, oxidative phosphorylation. And it all takes place on this microscopic but incredibly efficient energy production line called the electron transport chain.
Electron transport chain. Okay, that sounds kind of intimidating. Can you break it down for us?
Okay, so imagine it like this. Remember those, Christy? The folded shelves inside the mitochondria, embedded in those membranes, is a series of protein complexes, like a chain of workers on an assembly line. Each complex is ready to accept and pass along electrons, like a molecular bucket brigade.
So those NADH and FADH2 molecules are energy voucher holders, arrive at this assembly line with their high-energy electrons. What happens next?
As the electrons are passed from one protein complex to the next, they release a little bit of energy at each step. And that energy is used to do something really important. It powers these tiny pumps that move protons, those positively charged hydrogen ions, across the inner mitochondrial membrane.
So it's like using the energy from the vouchers to pump protons out of the matrix and into the inner membrane space. What's the point of that?
This creates what's called a proton gradient, a higher concentration of protons in the inner membrane space than in the matrix. Think of it like building up water behind a dam. All those protons are packed together, creating a lot of potential energy, just waiting to be released.
Okay, so we've built up this proton power. What happens next? How do we turn that potential energy into ATP?
That's where the star of the show comes in, ATP synthase. This amazing enzyme acts like a turbine, embedded in the inner mitochondrial membrane.
ATP synthase, a turbine. I'm intrigued.
It really is a remarkable piece of biological machinery. As those protons flow back into the matrix, driven by that proton gradient, they pass through ATP synthase. And as they flow through, they cause a part of the enzyme to spin, like a water wheel.
So it's like a tiny hydroelectric power plant. The flow of protons spins the turbine, which then generates power. But how does that spinning motion actually create ATP?
That spinning action provides the mechanical energy needed to convert ADP back into ATP. It's like attaching a generator to that water wheel.
Wow. So the whole system is powered by this flow of electrons, which creates a proton gradient, which then drives the ATP synthase turbine to generate ATP. It's a brilliantly designed system.
It is. It's a testament to the power of evolution and natural selection. But there's one more crucial piece to this puzzle. Oxygen.
Oxygen. What role does it play in all of this?
Oxygen is the final electron acceptor at the end of the electron transport chain. Without oxygen, the whole chain backs up like a traffic jam. The electrons have nowhere to go, and ATP production grinds to a halt.
So that's why we need to breathe. We're constantly supplying our cells with the oxygen they need to keep this whole process going.
Exactly. Oxygen is essential for life because it keeps the electrons flowing and the ATP synthase spinning. And there's a chilling example of what happens when this process is disrupted. Cyanide, that deadly poison, works by blocking the transfer of electrons to oxygen. At the end of the electron transport chain, it basically throws a wrench into the machinery, causing a catastrophic shutdown of ATP production.
That's scary. So no oxygen, no electron flow, no ATP.
Yeah.
And ultimately, no life.
Exactly. It highlights just how crucial this whole process is, you know. It's the foundation of energy production in most living organisms.
So we've got this amazing electron transport chain, powered by oxygen, churning out ATP. But what happens to all those byproducts? What about the carbon atoms from the original glucose molecule?
Those carbon atoms combine with oxygen to form carbon dioxide, which we exhale with every breath. It's a waste product of cellular respiration. And of course, water is also produced, another essential molecule for life.
This whole process is incredible. I'm still amazed that it's happening in every cell of our bodies, billions of times a second, keeping us alive and functioning.
It truly is a remarkable feat of biological engineering. And, while we've uncovered the main steps, there's still so much to learn about the intricate details and variations of this process.
This has been a fascinating journey so far. But I have one more burning question. How much ATP do we actually get from all of this? What's the final payoff from a single glucose molecule?
That's the million-dollar question, and the answer is surprisingly complex. We'll dive into the intricacies of ATP yield in the final part of our deep dive. Stay tuned.
Welcome back to Science Buzz. We're in the final stretch of our cellular respiration deep dive, and I'm ready to get to the bottom line. We've traced the journey of glucose through glycolysis, the Krebs cycle, and that amazing electron transport chain. But the question that's been nagging at me is, how much ATP do we actually get out of all of this? What's the final tally?
That's a question everyone wants to know, and the answer is, well, it's a bit more complicated than you might think.
Oh, really?
Yeah, you see, the exact amount of ATP produced from a single glucose molecule is still a subject of scientific debate and ongoing research.
Why, really? But we've known about cellular respiration for decades. Why is there still so much uncertainty? I thought it was like a simple equation.
It would be nice if it were that simple, but cellular processes are incredibly intricate, and many factors can influence ATP yield. The efficiency of the electron transport chain, the availability of certain molecules, even variations between different cell types, it all plays a role.
So it's not like baking a cake, where you follow a recipe and get a predictable outcome every time.
Exactly. For a long time, the classic textbook answer was 38 ATP molecules per glucose, but more recent research suggests that this number might be a bit optimistic.
Oh. So what's the current thinking?
Well, some studies estimate the actual yield might be closer to 32 ATP molecules per glucose, or even slightly lower.
Wow, that's a pretty significant difference. What accounts for that discrepancy?
Well, one key factor is the efficiency of those proton pumps in the electron transport chain. Remember how they use the energy from passing electrons to pump protons across the membrane? It turns out those pumps might not be perfectly efficient. Some protons can leak back across, reducing the overall proton gradient, and therefore the amount of ATP produced.
So it's like having a tiny leak in our hydroelectric dam, reducing the total power output. It makes sense.
Exactly. And there's another wrinkle. The transport of pyruvate into the mitochondria, remember that step from glycolysis? Well, that process also consumes some ATP, further lowering the net yield.
Okay, so there's no magic number. The exact amount of ATP can vary depending on a bunch of factors. But the bottom line is, cellular respiration is still incredibly efficient at extracting energy from glucose, whether it's 38 ATP, 32 ATP, or somewhere in between.
Absolutely. The precise number is important for scientific accuracy, but the key takeaway is that cellular respiration is an incredibly sophisticated and efficient process that's evolved over billions of years to power life as we know it.
It's truly amazing. From that simple sandwich to powering all our body's functions, it's a remarkable journey. And who knows, maybe one day we'll be able to fully unlock the secrets of cellular respiration and apply those principles to create new, cleaner, and more efficient energy technologies.
Now, that's a thought-provoking idea. Mimicking the elegance and efficiency of nature's energy solutions could lead to some groundbreaking innovations.
Well, this deep dive has been absolutely electrifying. I hope you've enjoyed this journey into the microscopic world of cellular respiration. If you enjoyed this video, don't forget to subscribe, share it with your friends, and hit the notification bell so you never miss an exciting new episode from Science Buzz.